Romain, the stage is yours, please.
Thanks a lot. So, hi everybody. So I'm very happy to be there today, and I would like to thank, indeed, Stéphane, Dirk, Michel, for the invitations. It's a good opportunity for me to talk about genetics and genomics. So don't be afraid. I will be very soft. I will try to explain how we can use genetics and genomics to study wild species and to study important characters in wild species. So, difficult after Aaron to discuss about wild species, so you know everything here. We have 130 species, maybe more, because we have more species growing the genus every years. That you have to know is we have mainly several phylogeographic groups, species from West Africa, like Robusta, from Central Africa, low and high elevations, like Arabica, Racemosa, for example, in the east part of Africa, and in Madagascar, with more than 66 species, Madagascar on Mascarenes Islands, that are a very high hotspot of biodiversity and hotspot of phenotypes, very interesting. This is just some example of the diversity of species here with leaves, different colors, shape, vein, domacia here. So we have Liberica, here the big leaves. And for the fruits, here it's more spectacular. So we have different color, shape, size of the seeds. For example, in the upper part, we have species from Madagascar, species from East Africa, Central Africa, and below from West Africa. So we have huge diversity, phenotypic diversity, and some years ago we put all this information in a database in order to share this information with public and researchers. This is a wild coffee species database. That group chemical information, geographical information, phenotypic information, and soon we hope genetic and genomic information in order to share the information to all people. Indeed, we have these species are a source of agronomical traits. As an example, we have the size of the seed here. We have two species from Madagascar, from the Coffea groups, Bicetiae and Boniensis, with huge variations of the size. We have also here resistance to disease and pests like rust, nematodes, and leaf miner in Racemosa, for example, and berry borer. It's a source also of interesting ripening time variations. So for Arabica, Canephora, it's more or less 9 months. But in the wild, with wild species, these variations range from 1 month of ripening time to 14 months, depending to adaptation to the environment. It is something that could be also to be studied to understand this adaptation of the genes behind these adaptations to adaptation to climate environment. And indeed, we have the adaptation to dry environment. Here is another example of a wild species from Coffea also. We have only 3 months of wet seasons. It's growing in deciduous forest, and we have very huge fruits here, and it's able to grow in this environment. And we have also a nice adaptation to seed germinations. The seeds can germinate 9 to 10 months after the ripening. Indeed, here it's a focus on caffeine. Caffeine also is quite important also for the industry. And we know that we have a huge variations of caffeine concentrations. So we have a phylogenetic tree here with the concentrations of caffeine and the phylogeographic groups. So mainly, we have no caffeine in Madagascar and Mascarenes Island, with the exception of 2 species, and the high concentration of caffeine are inside the phylogeographic groups of West Africa, like Robusta. And for the moment, these variations is still unknown. Why we have this variation? Why species in Madagascar don't need caffeine to grow? This is an interesting question in terms of evolutions. So we developed some applications in genetics and genomics in order to understand the evolutions and some traits of interest for the community. Here we have a phylogeographic groups with 82 species, or more than 200 samples, with species living in collections. These living trees. And we developed here, using short-read sequencing, a database of 10 million of markers. Here it's a restriction of 6 million, but with 6 million of markers, it's possible here to identify groups, identify species, identify hybrids, and to resolve some taxonomic challenge. If you have two species closely related, it's possible to separate and to identify. It's very powerful with 6 million to resolve everything. We apply this approach to groups in Madagascar, because it's a hotspot of biodiversity, and with 3 million of markers, it's possible to resolve the diversity here of species in Madagascar. And interestingly, our grouping is different from historical classifications used on botanical observations, indicating that this approach also is very, very powerful to resolve some challenge in taxonomy. These are applications with short-read sequencing, so just sequence of 100 base, for example. And we develop a new approach based on full genome sequencing. What it means that we sequence with another approach, with long read sequencing, like 10, 20 kbs. We reassemble all the genome, complete genome, and after we compare the genomes in order to understand gene variations and genes of interest, linked to trait of interest, for example. Here, we just a subset of our data, and we analyze 13 genomes, wild genomes, including also Arabica and Robusta. And some species are caffeine-free, another with low caffeine content, another with high caffeine content here. So we sequence everything, and we compare genomes. This science, the new science, it's called pangenomics. Pangenomics, it means that we compare genomes, compare chromosomes and genes, and we try to classify how many genes are in common and how many genes are specific. Here, for example, in the left side, we have the 11 chromosomes. We have two outgroups outside the Coffea in the upper side. We have 11 chromosomes, and we align chromosomes, and we compare how many genes are in common. This is called the core genomes in red. But the most interesting part are the green and the purple part. This is the dispensable genomes and private genomes. Private genomes, it means that we have only, the genes are only present in one species. This is a target of our analysis, because most of these genes here are involved in specific adaptations with new pathway of chemical compound, for example. Genes involved in resistance. And we are actually we are analyzing these private genes to understand the function of the genes and the correlations with environment, with climate, with presence of pest and disease. As an example of the pangenomics analysis, we apply this to caffeine, that is important for coffee here. So caffeine, we have here the metabolic pathway in the upper side. So it's mainly basics. It's very basic. It's 3 genes of the same family. It's methyltransferase that putting group methyls on a molecule. And you have 3 genes called XMT, MXMT, and DXMT. So the last one, DXMT, catalyzes the production of caffeine from theobromine. And the MXMT, theobromine from 7-methylxanthine. So it's very well known in Robusta. It's working like that. Okay. And we analyze these genes, presence, absence of these genes in this subset of complete genomes. including species without caffeine in blue, like Omole, Amblosiana from Madagascar, Racemosa, also from East Africa, Salvatrix from East Africa, Pseudozangue also from East Africa, and also Stenophila, Liberica, Brevicus, congensis, on Canifera. So in purple, you have low amount of caffeine in these species, and in red, a high amount of caffeine. And we analyze presence, absence of genes. This is the results here. So we have different genes here from the family, and the three last are XMT, MXMT, and DXMT. So indeed, if the three genes are missing, you have no caffeine. It means that in this species, we don't know why, the caffeine pathway is not present because the genes are not present. Very, very easy. In Amblosiana, also species from Comoros, only the last gene is missing. It means that it's not able to produce caffeine, and the pathway is stopping at theobromine. And we have a low concentration of theobromine in the seed. More interestingly, in Racemosa and Liberica, two species producing caffeine, but low amount of caffeine, the second gene is missing. MXMT is missing, but you have caffeine in the seeds. Low amount of caffeine. Why? This is exactly why here we are working on these species to understand why these species are producing caffeine if one gene is missing. This is a good example of the flexibility of the caffeine pathway. Even if one gene is missing, it's possible to produce caffeine, maybe with an alternative pathway, starting from 7-methylxanthine to caffeine. This pathway exists, I think, in tea, for example. Or maybe the DXMT genes is able, the protein, the enzyme, is able to have a dual functions. It has been shown also in Arabica. This enzyme is able to catalyze theobromine to caffeine, but also to use 7-methylxanthine and to produce theobromine. Maybe this explains why we have low caffeine content in Liberica and Racemosa. But also it illustrates also the use of genomics to resolve key traits of interest. So this was just a subset of the data. Actually, we have been funded to fully sequence 40 species. Indeed, we will focus on consumable and non-consumable species with low caffeine and caffeine-free species in order to understand the evolution of these species and to understand some key characters. And indeed, we will try to develop species-specific SNPs markers for identifications, also for improvement. You have some institutes that are making hybrids between Arabica and wild species. In this case, it's very important to have markers to follow the introgressions of key characters. And also, this project is important also to promote the conservation, so in situ and ex situ conservation efforts, especially in Madagascar. And this project involves 5 countries, 12 institutes, and if you are interested to join us, if you want to sequence and to explore genomics, don't hesitate to contact us. So, to conclude, what we have seen here is genetics and genomics are very powerful. Here we can resolve key characters, starting from here the presence, absence of caffeine, but also we can apply these techniques to other characters of interest using wild species and to understand the adaptation of wild species. And we hope that sequencing more species, more key species, we will be able also to understand the evolutions and the structural evolution of genomes and the functional diversity of key characters. Indeed, I would like to thank people that gave money, that's more important here. So the French Agency for Research, ANR, FAPESP, from Brazil, and the Agropolis Foundation, the Reform Foundation, and the groups involved in the British Coffea consortiums. And I forgot to put also Nestle, that's quite important, also that participate initially to sequence some genomes more than 6 to 8 years ago. So, thanks a lot.